Now that we've talked about impulses and how the action potential is actually generated, we can talk about about something with an impulse called propagation. Basically, the idea with an impulse and the action potential that we've talked about thus far is that it is local. In other words, it only takes place in a very small part of the membrane. But as we know, we have to be able to move this impulse along the neuron itself so that the information that it contains, which is basically that we have a stimulus that's arrived, can be passed to other cells, including muscles and other neurons. So how is this able to move along the membrane so that we can accomplish this? Well, there's a couple of different ways that this can actually move, and it's going to depend on a special feature of the neuron, of the axon itself. It depends on if the axon is myelinated or unmyelinated. So the first type of propagation we are going to look at is continuous propagation. Continuous propagation takes place on unmyelinated axons. So these are axons that lack the coverage with oligodendrocytes or with Schwann cells. Now, how does continuous propagation start? Well, the idea is that when we are at that peak of depolarization, okay, so that basically the number 3 on that chart that we just looked at, we have a lot of positive ions on the inside portion of the membrane where that action potential is taking place. But at the next segment right over from that, we still have negative ions. So we have this situation where on the inside in one segment we have positive ions, and in the other segment we have negative ions. We know from our basic chemistry that opposite charges attract each other. Okay, positives and negatives pull on each other. And so that's what happens. The attraction actually pulls the positive ions along the inner membrane surface towards that area where the negative ions are. That actually brings up the level of the membrane potential. So it goes from that negative 70 millivolts up towards the threshold of the negative 60, and we get depolarization to threshold, which in that next section of membrane generates a new action potential. So in essence, the action potential starts moving down the membrane in the next little section. This chain reaction, this progression down the membrane, little section by little section, continues forward towards the end of the axon. It's not able to go backwards because of something, the refractory period. Okay, basically the piece of membrane that has just generated the action potential, whose ions got pulled, is still in its refractory period. It's still actually restoring resting potential. So the new action potential can only go in one direction because the reverse direction won't respond. So essentially what happens is the action potential advances down the axon in tiny steps. But this process is very, very slow. It's only about 2 miles per hour, which may not sound very slow necessarily, but when we compare it to the other type, it is. So here is a diagram of what is happening. So basically in this first red section here, I get that depolarization to threshold and the generation of the action potential. A lot of sodium ions have come into the cell membrane, into the intracellular area of the axon. Next to this area, we have the negative area, and that actually pulls some of those positive ions down, which triggers the generation of another action potential in this segment of the membrane. And that just continues as we go on down the membrane. So that's continuous propagation. You can basically think of continuous propagation like drawing a line on a paper. You're not going to lift your pen or pencil off of the paper at any particular point in time. There's no breaks in it. And that's what happens with this action potential. There are no breaks in the membrane in terms of where an action potential is generated. Let's contrast this with the other type, which is saltatory propagation. Now, saltatory propagation, if you take Spanish, you might be familiar with the verb saltar, which means to jump. And that very much describes how this impulse is going to move down the axon. It kind of jumps. This happens on our myelinated axons. And what happens here is the lipids of the myelin sheath actually block the exchange of ions with the outside, where that axon is being covered by either the Schwann cell or the oligodendrocyte. So as a result, the ion exchange can only happen at those uncovered areas, the nodes. Now, the actual mechanism is going to look very similar. The positive ions move along the inner membrane surface, but they pull all the way down to the next node, and that's where the next action potential generates, is at the node. So the result is it looks as if the impulse jumps from node to node. Now, because you don't have to generate an action potential at every segment of the membrane at this point, this process is much faster. It can run anywhere between 40 and 300 miles per hour, so very fast. And so this is a diagram of what is happening. Okay, so we get an action potential generated here at this first part of the membrane. We get the pulling of those positive ions, not just to the next segment, but down to the next node. We get the exchange here of the ions, depolarization, and the new action potential get generated here at the node. And then again, the pulling of those, and then a repeat here at the next node. Now, the next video clip that I have is actually a really good one, and I do highly recommend that you go and take a look at this. It also talks about the combination of excitatory and inhibitory responses called summation that takes place. So now that we know how an action potential is generated and how it moves down the membrane, in our next segment what we will do is actually look at how action potentials move from one neuron to another.